I've been working on this disease since it started in March, and I've been looking at repurposed drugs to treat COVID-19. I just want to make it clear that the opinions in this presentation are my personal views as an academic research scientist. By way of background, in addition, I've 30 years experience working on clinical trials of antivirals, mainly on HIV, hepatitis C, over 200 published papers, Science, New England Journal of Medicine, The Lancet. But these opinions that I'm going to present today may not be exactly the same as UNITAID and the World Health Organization. The work I've been doing for them is to give them a proposal, to show them what's going on in the studies, to show them the results, and it's then their decision to take whether they would like to approve or take this forward. So the review from this, the results from this review have been submitted to those organizations for their analysis, but their analysis is still ongoing. So you shouldn't see any of this presentation as necessarily a signal or endorsement from WHO or UNITAID that this should happen. The results have not been peer reviewed, so should be seen as preliminary. So just to introduce this whole area, repurposing drugs for COVID-19, it's a strategy that's been pursued because you can take a drug with the known safety profile, with drug supplies that are already available in pharmacies or can be imported and made quickly, and the treatment can be adopted very quickly if the randomized trials show promise. So for example, we had the recovery trial of dexamethasone showing a survival benefit, and almost the next day, doctors starting to use dexamethasone in their clinics to treat oxygenated patients with COVID-19. The disadvantages, and I've experienced this at first hand, is you can think a drug is working for COVID-19 and that it can fail. You can get results that are promising in early trials, then fail later on. And I've worked on a number of studies of drugs, particularly sofosbuvir, clatosphere, the hepatitis C combination that looked promising early on and then didn't work. So one lesson I've learned is large randomized trials are needed to make clear assessments. So progress so far on repurposed drugs, we can see the benefits of dexamethasone giving approximately a 20% improvement in survival in the recovery trial for oxygenated patients. And then for intensive care patients, we have either tocilizumab or sarilumab, as shown very recently in the REMCAP-RAP studies in the United Kingdom. But we need to remember all of these, the drugs that have been approved so far are really for quite moderate to severe patients. There's nothing there for either mild patients, people not requiring oxygen, or indeed for prevention. There are other repurposed drugs that have been worked on that have not ended up showing survival benefits. So remdesivir, despite early promise, didn't. Lopinavir, ritonavir, used very early on in the epidemic in China, didn't show benefits. Interferon beta and infamously hydroxychloroquine. Other drugs I've worked on, favipiravir, nitazoxanide, losartan, imatinib, sofosbuvir, very much at an experimental stage and very mixed results from those other repurposed drugs. One thing I've learned with repurposed drugs is not to believe results from non-randomized studies, and everything I present in this talk is going to be from randomized clinical trials. This really is the gold standard of evidence that we need to assess any medicine for COVID-19. So just by way of background, what is ivermectin? There were many repurposed drugs being screened in the early phases of the epidemic in early 2020. Ivermectin was identified by a research laboratory in Melbourne in Australia as showing antiviral activity in vitro. But the IC50, the concentration of the drug needed to inhibit viral replication, was considered too high for in vivo activity to be likely. People were predicted to need huge drug levels or huge doses of ivermectin to achieve those IC50 concentrations. Now, as a result, this drug was not actively pursued in North America and Europe, and indeed Merck, the parent pharmaceutical company, have not evaluated ivermectin. However, given these IC50 results, some people believe that there was the potential for antiviral activity, and also it's an anti-inflammatory, and it might work through a different mechanism of action. So clinical trials have been started mainly in low and middle income countries where drug supplies were readily available. But when we think about this trial program, it's very different from the ones for the other repurposed drugs. We don't have the support of the large pharmaceutical companies. We don't have the support of the large donor organizations. There isn't a European or North American sponsor that can run the large trials. So you're really talking about a group of studies run with the resources available in the countries where it was set up. And one of the real attractive benefits of ivermectin, if it does work, a course of treatment of 0.4 milligrams per kilogram for 5 days already is available on sale in India and Bangladesh for $3 a kilogram of ivermectin costs $348 to make. So this could potentially be an extremely cheap drug. That's one potential benefit of it. It has huge huge safety profile from extensive use in other indications. So it's a widely available generic treatment being evaluated in 56 randomized clinical trials to treat COVID-19 worldwide in over 7,000 patients. The mechanism of action is likely to be anti-inflammatory based on animal models, but there are new estimates of IC50 emerging from human Calu-3 cells, as opposed to the original IC50 estimates that were from monkey cells. There's no individual clinical trial large enough to clearly establish efficacy, but the combined data from all the available clinical trials may be large enough to assess clinical efficacy reliably. So the research question we were trying to answer in this presentation was, is there enough clinical evidence to support the worldwide approval of ivermectin to treat COVID-19? And we looked at various different endpoints to assess efficacy, either viral clearance, time to clinical recovery, duration of hospitalization, or survival. So we conducted a systematic review of randomized trials of ivermectin to treat COVID-19. And this is a little different to the reviews that have been done previously. We did what other people have done. We looked at PubMed, Embase, the databases used to look at published information. The thing about ivermectin and a lot of repurposed drugs is the data is only available for some of these in preprint databases. So we looked at databases like medRxiv, Research Square. We also looked for all the clinical trials on clinical trials registry websites, and we contacted every clinical trial running of ivermectin worldwide. And this is something other people haven't done. We got their details, we asked them to join regular fortnightly meetings so that we could hear the results firsthand from the people running the clinical trials as they emerged. And throughout December and then now in January, we've been having these fascinating Zoom meetings with people literally from all over the world, countries like Japan, Lebanon, Bangladesh, India, all over South America, Israel, Turkey, just a whole range of different countries, all working in alliance and sharing the results from their individual trials. So the results I'm going to show you are from 18 randomized trials in 2,244 patients. And you can see here the range of countries we have: Bangladesh, Egypt, India, Iran, Iraq, Lebanon, Pakistan, Turkey, Nigeria, Argentina, and Spain. Now, this is actually a subset of all the clinical trials of ivermectin in progress worldwide. And you can see the 21 countries involved here. And actually, this list is expanding all the time. We're hearing about new studies being set up in some of these countries to further evaluate the results, the results we're seeing so far in much larger sample sizes. So in future, and I'll talk about this at the end, we should see results from 56 clinical trials, if they're all conducted, in over 7,000 patients. And even that number is going to carry on going up because we're hearing about new trials all the time. So what do we have at the moment right now? We have 9 randomized trials which looked at ivermectin as a single treatment on one day. And these trials generally look at the dose that's been used for parasitic infections, so 0.2 milligrams per kilogram, just as a single dose. And generally those studies have looked at ivermectin plus standard of care versus standard of care. So it's a simple add-on treatment. And you can see that some of these studies in the duration column there, the SB means single-blinded, the OL means open label, and the DB means double-blinded. So you can see these different designs, and quite a lot of the studies double-blinded, which is encouraging. Now, the patients included in a lot of the studies were mild to moderate. We had a few studies which looked at moderate to severe. And the sample size is generally quite small. We just need to be careful with these studies. We don't have the sample sizes in the range of the recovery trial or the solidarity trial. These are quite small trials, and some of these trials may not actually have the statistical power to show differences for all of the endpoints between the treatment arms. Then we have another 9 randomized trials, and these ones are looking at several days of dosing. Some of the studies in this category were actually dose ranging studies, so we can see the effects of different doses or different durations of treatment on the different efficacy endpoints. So you can see again a range of different countries: Egypt, Iran, Iraq, India, Bangladesh, Turkey, Nigeria, Pakistan, Argentina, really all over the world. And that's important when we look at the sources of bias. You can see the doses ranging from 0.1 to 0.2 milligrams per kilogram, right up to 0.6 mg per kg, and then going up to 5 days of treatment. So it's important to realize just the very large range of dosing we have, from 0.2 milligrams for 1 day up to 0.6 milligrams per kilogram for 5 days. So a 15-fold range of doses between the lowest and the highest. And again, some of the trials double-blind, some open label, and the patients a mix. A lot of the trials randomizing mild patients, and again the overall design of the study is generally to have ivermectin as an add-on treatment in addition to standard of care, which depended both for the country and the severity of infection. So more drugs being used for people with severe infection at baseline. There were a few studies that were head-to-head looking at ivermectin versus a different combination. So, for example, favipiravir as the control arm in the Turkish study by Okumuz et al. So the methods we're using in this meta-analysis, as I just want to emphasize, we've only used randomized clinical trials. In the WHO grade criteria, a systematic review and meta-analysis of randomized clinical trials provides the highest level of evidence to support recommendations on treatment. Now, we've really concentrated on survival as our primary endpoint, and for that, there's this technique of Cochrane Mantel-Haenszel testing with inverse variance weighting, random effects modeling. It's a standard technique that's used in meta-analysis. We looked at other outcomes summarized for each study individually, and we combined them when we could. Other groups are looking at combining these also, and it would be much better to have the raw data to back up these analyses when it's available. Now, because we have this wide range of doses, we looked at effects of ivermectin dosing on response as well. Now, in any meta-analysis, it's important to look not just at the results, but the quality of each clinical trial. So we followed what are called the Cochrane criteria. The Cochrane analysis is a standardized form of systematic review. There were guidelines in 2011, updated in 2019. We've looked at both systems, and it really comes down to how reliable is the evidence coming from these studies. So first of all, what's the method they use to randomize patients? Has it been explicitly described? Are the arms comparable at baseline? Was the randomization successful? Were the investigators and patients blinded? If in a double-blind study, nobody knows whether they're taking the drug or placebo, and that makes the study more reliable because you can judge endpoints objectively. There are some endpoints which are not so affected by this issue. If, for example, survival or a readout from an assay machine such as viral load, those are called objective endpoints, and they're not as prone to bias. How complete is the outcome data? Is there a risk of attrition bias where you lose more people in one treatment arm than another? Or is there selective reporting of outcomes? So we really interrogated these trials to see whether there are any sources of bias. And as I said, when we look through these results, we need to look at objective outcomes where they basically are apparent whether somebody's blinded to treatment or not. So whether somebody's alive or dead, you can't change that by clinical judgment. And the result coming from a PCR machine looking at viral clearance. Those are called objective outcomes. But subjective outcomes, if you know what treatment is somebody's on, there's the potential to make assumptions about the treatment and its effect. So if you know that somebody's taking an active drug, you might think that they've clinically recovered more quickly or they're ready for hospital discharge. So we've put less weight onto those endpoints. So let's start looking through the results from this meta-analysis. Now, this is data that hasn't been shown before. So ivermectin could be working as an anti-inflammatory as well as an antiviral. That's what's apparent from the animal models of ivermectin in Syrian golden hamsters. So when we look through the studies, first of all, you can see here the Egyptian study, Elgazzar, we see significantly greater reductions in C-reactive protein for ivermectin versus control. In the same study, these are for the severe patients, we see significantly greater reductions in ferritin, and we see significantly greater reductions in D-dimer. In the same study, for the patients who are less advanced, there were not significant reductions, but we can see this effect for the more severe patients at baseline. If we look at the same information from the Iranian study, and you can see C-reactive protein here in the blue for the placebo arm remaining constant, and then for the people taking ivermectin at different doses, different number of days of treatment, you can see reductions in this other marker, C-reactive protein. Then the same information from the study in Bangladesh, you can see C-reactive protein significantly falling for people taking ivermectin for 5 days, and borderline reductions for people taking ivermectin, borderline significant reductions in ferritin. Whereas if people are taking the drug only for a single day, the reductions in C-reactive protein are then borderline significance, and there's no significant change in ferritin. So there's evidence here for dose-related reductions in inflammatory markers. So viral clearance is an endpoint that's used very widely in studies of mild infection. It's not been evaluated in all of the countries because some countries had very limited supplies of viral load tests early in the epidemic. I worked a lot with centers in Iran where they had very limited access to viral clearance, to viral load assays. So we'll see this for some of the studies. Now, when we look at the effects of ivermectin when it's given at a relatively low dose for a single day, we do see significant benefits in the larger studies. So you can see in Bangladesh, people taking 12 milligrams for a single day, 8% were detectable after day 14 versus 20% on the control arm. A highly significant difference, shows an antiviral effect. And similarly in Pakistan, 90% versus 44% were undetectable by day 7, so another antiviral effect. We have other studies where if the patients are very mild at baseline, and you only look at a long-term endpoint, you look at day 10, then you don't see a difference between them. And that's really because by that time, everybody taking control has already shown viral clearance, but you don't have the ability to see early effects. You've looked too late. And I'll show some data later on which shows how if you look earlier in the course of treatment, you can see effects that you then miss if you look later on. In the Lebanese study by Hussam Murad, we saw effects on the cycle time values, statistically significant. And then in the Indian study, Mohan, there was a trend, but it wasn't significant. We need to remember with some of the smaller studies, we might not have the statistical power to see the differences between the arms. But remember, these first studies I'm showing you are looking at a single day of treatment with relatively low doses. Now, if we look at dose ranging effects, you can see here for the study in Bangladesh, when ivermectin is given for 5 days, you get a significantly greater proportion of people showing viral clearance by day 14, and that's in the green, with a p-value of 0.005 versus placebo. If you give ivermectin for only 1 day, you don't see a significant benefit over placebo. So there is evidence again for dose-related faster viral clearance with ivermectin, so 5 days better than 1 day. If we then look at the data from Nigeria, You can see here you have in red people taking the higher dose, showing a slightly faster rate of viral clearance. And it shows here how important it is to look very early in the course of treatment to see these effects on viral clearance. You can see in the green, people on placebo generally being slower to clear the virus. And here in the Nigerian study, we had a significantly faster rate of viral clearance for the 12 milligram versus placebo, double-blind study. So we can see quite consistent effects between the studies on a dose, with the correlation between dose and viral clearance. And finally, in the Argentinian study, this was what's called a PK/PD analysis, where you look at the concentration of ivermectin versus the speed of viral clearance. And you can see in green, the people who had the highest concentration of ivermectin showed the fastest rate of viral clearance in this small study. So we have results from various different studies suggesting a dose response. So then if we move on to the studies of ivermectin that we're looking at the higher doses for longer durations of treatment, this is where we start to see the really significant effects. This is where we see 50% faster viral clearance. So in the Egyptian analysis, 5 days versus the people on ivermectin becoming virus negative after 5 days versus 10 days on the control. And similarly in the severe patients becoming negative after 6 days versus 12 days. And we see this effect really throughout a range of studies. The Kirti study didn't systematically evaluate PCR in all patients. They were missing a lot of data, so it's hard to evaluate that particular study. The Turkish study, again, was quite a small subset, but it was actually statistically significant. In Argentina, as I was saying, we had this dose-related effect on viral clearance. In Nigeria, the people taking the higher dose became negative in 6 days versus 9 days on placebo, p equals 0.003. And in Bangladesh, people taking the drug for 5 days became undetectable within 10 days of randomization versus 13 days in the placebo arm with a p-value of 0.005. I've been working on the repurposed drugs right since the beginning. I haven't seen any data like this so consistent between such a wide range of countries, looking at the data in slightly different ways, but getting quite consistent conclusions. So you can see here the graphs of time to viral clearance, and basically a shorter bar means they get the virus undetectable more quickly. So 5 days versus 10 days, 6 days versus 12 days, 9.7 versus 12.7 days, and 6 versus 9 days. So 4 different studies, 3 different countries, and significant p-values across the board. Let's move on then to clinical recovery and hospitalization. Now, what we need to bear in mind with this analysis is that there is some subjectivity in how it's measured. People might believe that somebody's clinically recovered earlier if they know they're taking an active treatment. So we need to be a little bit more circumspect about how we interpret this data. So you can see, first of all, again, I've grouped the studies for those given for 1 day versus multiple days of treatment. And we can see that among the studies looking at 1 day, we do get effects. But when we look at how large they are, what we're talking about in terms of clinical recovery, maybe a day earlier, days in hospital, maybe 1 or 2 days fewer. So we're getting significance, but it's not a huge treatment effect. If we then look at the studies with greater doses and longer durations of treatment, this is where we see the bigger effects. And they're not significant in every study, but sometimes that's an effect of sample size. But we're getting generally a more pronounced effect on hospitalization and clinical recovery, where we use larger doses and for longer periods of time. Now, the approval of dexamethasone, tocilizumab, sarilumab was really based on survival. Now, when we look at survival, we now have results from 6 different studies. Now, this is different to the analysis that was put on YouTube, who actually allowed that to happen. But if we look at these 6 different trials, most of the studies are using multiple days of treatment. There are very few. There's one in Bangladesh that looked at 1 day of treatment. And you can see across these different studies, you get these trends where fewer people are dying on ivermectin versus control. You can see that in Iraq, in Egypt, in Bangladesh, in Iran, in Turkey, in India. And if you put the results together, 14 out of 658 people taking ivermectin have died during these studies versus 57 out of 597 taking control treatments, a difference of 2% versus 10%. If we then do a standard forest plot where we look at the relative risk of dying for ivermectin versus placebo, you can see in each study there is a trend for fewer people dying. And then if you add up all the results, what you get is a 75% survival benefit with confidence intervals of a 48% survival benefit to an 88% survival benefit. There's also an important number here which is called the I-squared, and this shows how consistent the results are between the different studies. A 34% I-squared means that it's a moderate level of heterogeneity. So there might be some differences between the studies that might actually be explained by some of the studies recruiting more severe patients, such as the Cochrane study, where you do get people dying on any treatment. Some people can die very quickly. So that might explain some of that heterogeneity. So what are the limitations of what I've just shown you? And I think we need to be very careful when we look at all this data that we don't get carried away, because at the moment we have data from 18 randomized trials in around 2,167 patients, but that is a subset of all the clinical trials that are registered. So are we in the situation where it's looking good so far, but do we need to check the results coming out in the future? There is a potential for publication bias. We might be seeing the trials which are looking good, but we've made every effort. We've reached out to the studies all over the world, and we've had these international meetings. We've really tried to find every trial result, whether published or unpublished. There might be more trials with unpublished results. We simply don't know about them. When we've been looking at these trials, we've found generally that the trials with unpublished results are simply ongoing. There are some studies, for example, in Australia, where the epidemic never really happened. They couldn't recruit. The same in Thailand. We've got 3 studies in the United States I don't believe have ever started, but we need to get confirmation. And then there are study programs in Egypt, in India, in Iran, which are ongoing. We need to see the results as they come out. We need to remember when we look at individual trials, they can have limited statistical power. Several of the trials are open label, so there is this potential for investigator bias. And at the moment, we're looking at efficacy, but we have this very wide range of doses and durations, and the endpoints could differ between the trials. We've looked at what's called the risk of bias assessment. So we've looked at all the different ways that these results might be affected by how the trials were conducted. And we need to remember there are some potential issues with the studies. We don't have the raw databases. We're working to collect them at the moment, all the documentation, and the quality of the evidence in the Cochrane reviews is, when it says limited on the right hand side, that refers to the primary endpoint used in the study. Now, if the primary endpoint was hospitalization or clinical recovery, and the patients, people knew what treatment they were taking, there's the potential for bias. But when we've repeated this same risk of bias assessment looking at survival, it's very hard for survival to be affected in an open label or a single blind study. There are some other issues. Some of the trials didn't have complete follow-up. They lost some of the patients, people lost to follow-up. But when we've looked at that, we haven't found differences we haven't seen evidence for different rates of loss to follow-up between the treatment arms. There was a study in Iran where people were diagnosed based on CT rather than viral load. That's very standard in Iran because they have limited access to the assays, and they're very good at the diagnosis. They were one of the first countries to suffer an epidemic. So what are the implications of these results? The first one is, we know from the treatment of HIV that treatment as prevention can lower the risk of transmission. U equals U, undetectable equals untransmissible. That is a hypothesis for COVID-19. And it's quite possible that if we can lower the viral load faster, get people cleared faster, those people might be less infectious. We're seeing this treatment effect consistently across randomized trials. And if the undetectable viral load lowers the risk of transmission, there is the potential to repeat the success story of HIV test and treat for COVID-19. I'll just show you how that might work. So you could imagine a situation where you had a rapid viral load test. People who are tested negative could either be offered a vaccination if they're available, a preventative strategy, and then the people who are tested positive, they would be given treatment with ivermectin potentially to make them less infectious. And there are studies which are of ivermectin actually as a preventative strategy. So you can see here we have 3 studies, 2 conducted in Egypt and 1 conducted in Argentina, showing that people who take ivermectin, in these randomized studies only, having lower rates of infection for ivermectin versus control. So we have a lot of interesting ideas that could be pursued in the test and treat arena. So far in this meta-analysis of 18 randomized trials, so far we're seeing associations with viral clearance, duration of hospitalization, rates of clinical recovery, and a 75% improvement in survival rates. And we're seeing these effects for viral clearance to be higher for higher doses and for longer durations of treatment. Now, before we get too carried away, we just need to remember that this database at the moment may not be large enough or robust enough to allow immediate regulatory approval. We shared these results with the National Institutes of Health in the United States, and their impression was that this is all data based on preprints, on personal communication with investigators, and really they need to see more documentation. They need to see the actual databases, the protocols, all the patient data at an individual level to be absolutely sure. The drugs are not approved based on information that goes into preprints. They're approved based on submitted quality data. So the next steps from the team's point of view is this very detailed assessment of data quality from each trial. We're using the Cochrane criteria, evaluation of bias. We're integrating results from emerging trials, and there are many other studies generating results, and they will be reporting in the near future. So the way that the WHO indication works is you have a separate indication for mild infection to lower the risk of hospitalization and to cause faster viral clearance, and then you have an alternative moderate severe infection indication where the endpoint is faster recovery and improved survival. Now, as I said, we've seen data so far from just over 2,000 patients. We have a lot more data coming in the next month, and we just need to be a little bit careful here and remember what has happened with previous trials where remdesivir looked great until we saw the results of Solidarity. We have more trials coming, and we need to consider how much we need to wait for these results before we make very big decisions on approval. Bulgaria, 120 patients, Brazil, 700, Colombia, 450, another study in Colombia is 66, Argentina, 500, all arriving with data within the next 4 to 6 weeks. And at our regular meetings, we'll be seeing these. The Mexicans are just about to finish a 3,000 patient dose ranging study where they've looked at different courses of treatment, either for 4 consecutive days or with the doses spread over a week. We'll have a good safety database from Mexico, all coming in about 4 weeks from now. In March, we'll then see results from 2 Brazilian studies. All these studies are randomized, 800 from one, 400 from another. People are going to higher doses. They're looking now at 1 milligram per kilogram up to 5 days. There's an Italian study looking at 1.2 milligrams per kilogram, so much higher. There are also clinical trials in progress in Egypt. In India, there's a 1,800 patient study in Iran, which could have results emerging in April. But we need to remember, when we're trying to predict the timelines out 3 months from now, the epidemic is very hard to predict, and you'll get countries, I'm sure you remember in South Africa when the first wave disappeared and you couldn't recruit studies anymore, and then suddenly the second wave appears and you can recruit them. And the Egyptians are on the downslope of their current wave. They're finding it hard to recruit. So we need to be careful predicting timelines for the later trials. Now, I just want to stress, this is my personal opinion throughout this presentation. the question is coming up, when to approve ivermectin. Now, we had data in December from 1,452 patients, 11 trials showing an 84% survival benefit. Now in January, we have 18 trials in 2,294 patients showing a 75% survival benefit. In February, we'll have data from 23 trials in 4,100 patients, and so on, going up to April, where we get to 10,000 patients. Now, there is clearly a dilemma with early decisions versus late decisions, and we've seen people rush into approving hydroxychloroquine or various other treatments, remdesivir, too quickly. And just like with those other drugs, if you were to approve a treatment right now, one thing we're facing in the UK, we don't have any drug supply. We don't have ivermectin in the United Kingdom. I don't know what supplies you have available ready in South Africa. And then there's the added danger of what if the new trials do not validate the current results? What if we've just been lucky? And I think that's unlikely, but it's possible. And then the converse is if people delay the decision for too long, if you wait for another 3 months, a lot of these repurposed drugs, they simply sell out worldwide. And it could be that if nothing's done, the supplies could simply disappear and there'd be no ivermectin to get hold of. The other issue is that if it was delayed, then you need to count the cost of not treating people who otherwise could have benefited. Now, one option that I've been suggesting to people is what's called at-risk drug ordering. And this was done for the vaccines. So with the vaccines, the British government, other governments, they predicted that the vaccines would be efficacious, that they would be preventative. And so they did what's called an at-risk order. They said to Pfizer and AstraZeneca and Moderna, here's an order for the vaccine. We don't know if it's going to work, but we're going to put the money on the table and we're going to order it. And then if it works, it means we can deploy it immediately. So there's the potential for, in the same way, at-risk drug ordering for drugs that look the most promising. Now, even if the drug doesn't work, there are probably other countries that could use it to treat parasitic infections, filariasis, onchocerciasis, strongyloides, etc. So it's not money that would disappear, but it's a way to secure a supply of drugs so that if the results emerge looking efficacious in February and in March, the drug would actually be available to be used. So with this option, it will be possible to make a decision on approval when the evidence reaches whatever threshold for reliable conclusions that each regulatory authority wants to consider. Now, I don't know if at this point I should just directly answer these questions, but I was sent some questions in advance to answer. So when will the meta-analysis data be released? We've put the paper on a preprint server. It's just that the preprint servers, they have various questions you have to answer before the report is online. So once those questions have been answered, formatting questions, then it will be released, but we're talking at just a few days. So I was asked the question, would you recommend a family member in hospital with COVID-19 to enter a placebo-controlled trial of ivermectin? So I'll give you an example. My brother, he's 58, he's a smoker, and if he was hospitalized with COVID-19 and there was a placebo-controlled trial in the UK, that would be his only option to get the drug. So I would say yes, go into that study. I don't know that there isn't a way to get supplies of ivermectin into the UK. If I could, and I knew it was good quality, I would want my brother to be taking it. In a court of law, would you be able to claim that ivermectin was clinically effective? I think we need to look at the level of evidence that we have at the moment. All I would do is repeat what I've presented so far and say that we are at an interim preliminary stage. We have the results already from 2,200 patients, but we need to remember we have more data arriving. We need to be a little bit careful that we don't get ahead of ourselves and assume that we're going to see this 75% survival benefit in the long term. There are confidence intervals around that survival benefit, anywhere between 48% improvement in survival and 88% improvement. Even if we hit 48%, which is the worst case from the current analysis, that would still be a major advancement in treatment. How about other co-treatment with other drugs in the trials? This is a comment from the US National Institutes of Health. It's a very fair comment. The ivermectin was generally given in addition to a standard of care, and the standard of care does differ between the studies. And we will be showing data on how many people were taking different parts of the standard of care, bearing in mind that some of these studies were run quite a long time ago, where the standard of care did include drugs like hydroxychloroquine and lopinavir that are no longer favoured. Could ivermectin affect immunity post-COVID? I think that's a question with no answer at the moment. We haven't seen results on the evolution of antibodies. Does ivermectin have an effect on hospitalization? I have the impression that this is dose related. We need to be careful about the evaluation of bias and when we look at this endpoint, because it could be affecting the open label studies by knowledge of treatment, but there are significant effects across a range of studies on hospitalization. Are there other factors such as dosage which could affect the efficacy? Clearly, yes. So the rate of viral clearance is faster for people taking ivermectin at a higher dose, up to, we have experience up to really 0.4 milligrams per kilogram and up to 5 days. Other new studies are looking at 1 milligram or even 1.2 milligrams for 5 days, but that is then outside the safety database for ivermectin used to treat the normal parasitic infections. Can these data be used to accelerate the acceptance of ivermectin as a treatment for COVID-19? Absolutely. This is why we've done the analysis. But I just need to stress these are my personal analyses of the data, and it's up to the regulators to make their decisions, and it's important to contact them independently for their review, their opinions. What importance should be placed on observational studies and unpublished studies? Now, I've had a lot of problem with observational studies in COVID. There was a case control study done of remdesivir, published in JAMA, I believe, which showed a 63% improvement in survival. And it was a press release, it was widely reported. And then in Solidarity, we saw nothing. The original observational study of hydroxychloroquine from the French group, Didier Raoult, showed an effect of hydroxychloroquine, which was never seen. So personally, it sounds quite harsh, but I'm just not including any observational studies in our analysis. I'm relying on just on randomized studies. Unpublished, now the problem with unpublished studies is that they haven't been peer reviewed. But if you think about the approval of dexamethasone through recovery, it was an unpublished study at the time. The approval of tocilizumab through REMCAP-RAP, again, it was an unpublished study at the time. So we are working, like it or not, with COVID, we're working with unpublished studies. The people running the studies of ivermectin are finding it very difficult to get their results published because people don't believe in the drug. And it's really a vicious circle. We keep going round and round. There's no data published, so people don't believe it. You can't get the new data published. I think we need to break that. And actually, the publication of a trial in the Lancet today is a major advance. So we need to get this data into the public domain. Methodological issues with the Elgazzar trial. So we did a sensitivity analysis of survival, and we took out each one of the studies. So we had 6 trials showing a 75% improvement in survival. Even if we took out the Elgazzar trial, we still saw a significant effect on survival. So it's not driving the analysis. What can we learn from other repurposed drugs in terms of bias? I think we need to, I keep saying it, we need to keep with large randomized trials and systematic reviews and meta-analysis, and not work from pilot studies that show promise that can then disappear. So I've answered all the questions I was asked, and I'd just like to thank you very much for coming, and I'd be very happy to answer any more questions within the time allowed.